Good afternoon, everybody. Welcome back after the break. I hope you enjoyed the field trip if you were joining Hilary and Jack there. Welcome to the start of session 13B. In the interest of time, we're going to start pretty promptly. So I'm going to hand over to our first speaker, James Charles, who's going to be talking to us about the evolution of bipedal locomotion in humans. So I'll just hand over to James. Super.
Right, thank you, Lauren. As she mentioned, I will be talking about foot anatomy, walking energetics, and how all this links to the evolution of bipedal locomotion. And this is part of my postdoctoral work at the University of Liverpool. So, in terms of the efficiency of locomotion, the variation of this between various species is fairly well studied, with notable research suggesting that small animals are actually relatively inefficient relative to larger ones. However, it is this variation within different species and the reasons behind this which is less well studied and therefore less clear. However, the research done in humans, the research of that which there is, usually looks at running. However, there is relatively few studies into the cost of walking. And this is somewhat surprising given the fact that walking is the most common form of locomotion in modern humans, as well as the fact that this is likely to link quite strongly with studies into the evolution of human bipedal locomotion. However, what these studies have found is that the variations within modern humans in the energetics of locomotion are likely to be somewhat related to various aspects of the anatomy of the lower limb and foot. So the modern human foot and ankle complex is fairly unique amongst us, our immediate ancestors in hominins, and our great ape relatives. This is because we have a unique combination of a pronounced medial longitudinal arch as well as transverse tarsal arches, a long Achilles tendon which attaches to a large robust calcaneus bone, as well as long lower limbs. And it is this suite of features which as I mentioned, are unique to us and help to optimize our efficiency of bipedal locomotion. However, the functional effects of many of these traits in isolation are unclear. And this is particularly so over different types of terrain. And this is because the research that has been done into the energetics of human locomotion usually do so over flat level surfaces or using various different types of treadmill. However, compliant terrains such as mud or sand, for example, are known to increase the energetic cost of locomotion or the cost of transport quite substantially. However, exactly how variations in various anatomical factors, such as the ones I mentioned above, mediate this increase in cost of transport is unknown. And this is particularly important for the study of human evolution, as knowledge of how anatomy affects functional behavior in modern humans could give more insight into these interactions in hominins. And furthermore, how or if the modern human foot is evolved to optimize the efficiency of walking over different surfaces. So the overall aims of our study were to investigate interactions between lower limb and foot anatomy, metabolic energy expenditure in modern humans during walking over different types of terrain. And this will allow us to answer questions surrounding functional impacts of anatomical variation in the modern human foot, as well as if the modern human foot anatomy is specifically adapted for walking over compliant terrains. So this was addressed with an overarching hypothesis which stated that based on previous literature, which suggested weak or no correlations between lower limb and foot anatomy and walking energetics over hard level surfaces, that our data would show similar results, and that there would be little correlation between lower limb and foot anatomy and the energetics of walking on a floor. However, we did predict stronger correlations with the cost of navigating more compliant substrates. So to quickly go over our experimental setup, we had 29 young healthy individuals who we asked to walk for an extended period of time over 3 different types of terrain or surface. So this included a hard level floor as well as a foam of 2 different thickness and compliance. And we selected these surfaces as proxies for various different types of terrain which may have been encountered by hominids. And while they walked over these surfaces, we measured their oxygen consumption, and then from this we calculated the cost of transport of their walking. We also asked each subject to walk over a pressure sensing treadmill to generate average footprints. So we then regressed this cost of transport data against various anatomical factors such as lower limb length, calcaneus length from MRI, foot shape, which is from the pressure treadmill, and this was the ratio between midfoot width and forefoot width, as well as ankle plantar flexor strength, which was directly measured from an isokinetic dynamometer. And it is this calf muscle strength which is thought to be related to the ability to stiffen the foot during walking, so it's therefore likely to be related to the efficiency of walking. So for our initial results, we found that over the more compliant foam substrates, cost of transport increased significantly relative to walking over the flat level floor. And in terms of variation within our study population, we found that there was quite substantial variation in terms of the cost of transport within our study population over each surface. However, in terms of the variation in anatomical parameters, we found that lower limb length and calcaneus length in particular were not particularly variable between our study population, suggesting that the subjects that we chose for the study were actually quite homogeneous in terms of some aspects of their anatomy. However, ankle plantar flexor strength and foot shape were far more variable within our population. So before I move on to describe our regression results, it should be noted that we initially regressed everything against body mass and found that pretty much everything, so this includes each anatomical factor as well as cost of transport, pretty much everything was significantly correlated with body mass. So we then took the standardized residuals of these regressions against body mass and used them in further analyses. And this essentially just normalizes everything to body mass and removes that as an effect in the study. However, even after accounting for body mass, we did not find any significant correlations between any of our anatomical variables and the cost of transport of walking over any surface. So this includes the floor, which agrees with our initial hypothesis that there would be no relationship and previous data. However, the lack of correlation on the compliant foam surfaces just goes against our original hypothesis. And this applies to, as I mentioned, each anatomical parameter. So this is for lower limb length and calcaneus length here, but it also applies to ankle plantar flexor strength and foot shape, in which we also found no significant correlation. So, as I mentioned, these results do not support our initial hypothesis that there will be a significant relationship between morphological variation in lower limb and foot anatomy and the efficiency of walking over compliant substrates. So despite the high intraspecific variation in cost of transport over all of the surfaces we studied, this does not appear to be explained by variations in the foot anatomical parameters that we studied. However, this is interesting in the context of human evolution, because the cost of transport on the floor does not appear to vary particularly greatly from values in the literature predicted for earlier hominid species. And this is despite fairly large differences in various anatomical parameters such as lower limb length and calcaneus length. So this, in combination with our experimental data, casts a certain degree of doubt over the functional significance of foot anatomy and lower limb anatomy in modern humans for determining the efficiency of walking. And perhaps more pressingly, this casts a similar degree of doubt over the functional significance of small variations in musculoskeletal anatomy in the fossil record. So, however, it is likely that locomotor efficiency is determined by factors beyond musculoskeletal anatomy. So this includes posture, kinematics such as joint angles, as well as the center of mass movement during walking and various other dynamic movements for producing inverted pendulum motion. Muscle physiology such as muscle fiber types, as well as tendon mechanical properties such as tendon stiffness, given that tendons are known to store a lot of elastic energy during locomotion and therefore contribute greatly to the efficiency. And we plan to use musculoskeletal modeling and computer simulations in future work to answer these and other questions. So musculoskeletal modeling is a platform by which you can predict things which are hard or difficult to measure in an experimental context. And we will use this platform to simulate walking over our foam surfaces and predict muscle functions, muscle mechanical work, and other factors in each individual. and relate this to variations in musculoskeletal anatomy, cost of transport, etc. It's also important to address some limitations of this study. So we looked at walking, which is a fairly low demand activity despite being applicable to the study of human evolution. And as I previously mentioned, we had a fairly homogeneous study population in terms of a lot of our anatomical variables. So we can improve on this in further studies by looking at running, for example, which is a high demand activity, so therefore might introduce some more variation in terms of the cost of transport to our data set. But we can also measure similar or indeed other foot anatomical metrics in different populations over more natural terrains. So this will only serve to increase the applicability of our results to the study of human evolution. So in conclusion, as the take-home messages, we can say that our data suggests that lower limb anatomy and foot anatomy does not appear to affect the efficiency of walking over compliant or indeed non-compliant surfaces, in our modern human population at least. So therefore, the importance of small variations in musculoskeletal morphology in the fossil record should not be overstated in the functional context. And this not only applies to human evolution, of course, but also potentially paleontology in general, when trying to infer the functional capabilities of an extinct species. However, we do recognise that further work is needed to fully investigate this, such as I mentioned before, computer modelling techniques, looking at different populations during high demand activities such as running, and looking at things over more natural terrains. So that's been done, so I want to thank you all for listening. I want to thank my colleagues at the University of Liverpool and in the Evolutionary Morphology and Biomechanics Lab and the Liverpool Trust for funding, and I will happily take any of your questions.
Super, thank you very much, James. It was a brilliant talk. So the floor is now open to questions, of which we do have a few in the chat here. So first of all, I'll just go to Fabio Alfieri, who says, brilliant talk, really enjoyed it. But he wants to know whether you took into account robusticity in the lower limb long bones.
Well, we didn't for this study, and that's a very good question, but we do have a whole suite of medical imaging data from these participants. So we took 3 different types of MRI from them, so the foot MRI as well as the long bone MRI. So that is something we can definitely look at in the future.
Super, excellent. I've got another question here from Kristiaan Janis, who says, how did stride length vary with the surface? Because that will obviously affect the COT.
Yeah, that's also a really good question, thank you. And also, so I'm working with a PhD student on this project, and that is sort of her side. So I don't actually know the answer to that question, but because she's looking at
someone does.
Stuff. Someone does,
yeah.
And it will be in the next paper.
Brilliant, thank you very much. Good, nice to see that research will be kind of coming out then. So a final question from the chat here from Stephen Hurrell. So actually asking a similar sort of question. Does the stride length vary according to the softness and hardness of the material? So I guess that's also something that this PhD student will be working on in the future.
Yeah, so I could just expand upon that a little bit. So I actually took part in the study, and I think I can say that stride length, initially I can say that stride length did increase.
Yeah.
Because I remember doing it. But also, yeah, I think stride length is something that we have briefly looked at and found an initial correlation.
Super. I'm going to exploit my position as chairing and ask a question of my own. So at the end there, you said that you were going to look at running, and given that foot shape is one of the kind of important things that actually does kind of vary a little bit, would you perhaps expect to see a greater impact of foot shape if less of the surface of the foot is being used?
Yeah, hopefully. So
the feet.
Yeah, so that's a really good question, and actually a follow-up study we've done is on sand, where we've looked at footprints. So that
fabulous.
Can also, be a better way of looking at that. But yeah, no, I sort of hoped to find a good correlation with foot shape, because there's an awful lot of data out there on how flat foot, high arch, or whatever actually impacts locomotor efficiency. But
well, that'll be your moneymaker eventually, right? Gait analysis. Okay, super. Well, thank you so much, James. We're going to move on to our next speaker, but I'm sure everyone will get in touch in the chat if they have further questions. So next up, we've got Sophia Anderson. So Sophia's going to talk to us about bat-winged dinosaurs and whether or not they could fly. So please take it away.
Hi everyone, so my name is Sophia and thank you for joining me to talk about the bat-winged dinosaurs. Now, I'm just going to move myself out of the way there so you can see this. The Scansoriopterygidae are a family of theropod dinosaurs from the Yanliao biota in what is now Hebei province, China, dating from about the mid to late Jurassic, approximately 163 million years ago. The family contains 4 genera, which are shown here, and they have been confidently placed within the Maniraptora, which is a theropod group including birds, dromaeosaurs, and their close relatives. But beyond that, their exact phylogenetic position is as yet unresolved. There are 3 potential phylogenetic placements for the Scansoriopterygids, as basal birds, basal Oviraptorsauria, or basal Paraves. The key thing is that from the very first description of Epidendrosaurus in 2002, discussion has arisen surrounding their elongated forelimbs. But when E-Chi and Ambopteryx longibrachium were discovered with a unique styloform element and a forelimb membrane, the debate only grew stronger. It's very clear that the Scansoriopterygids were experimenting with unusual forelimb morphology. But the question is, are these wings? Could these animals achieve powered flight? Well, first we need to define exactly what we mean by powered flight. This refers to remaining volant once you're already airborne, usually via flapping behavior. And that's in contrast to gliding, which is another volant behavior that takes the form of a kind of controlled aerial descent. Gliding has arisen many, many times across vertebrates, including rhacopherous frogs and colugos, but powered flight has only arisen 3 times in birds, bats, and pterosaurs. So how does the anatomy of Scansoriopterygids compare to that of other vertebrates? Well, despite being related to birds, Scansoriopterygids did not have bird-like wings. All 4 genera have been found with associated feathers, but never with pennaceous or flight feathers. Those are the feathers that allow birds to fly, and they are necessary but not sufficient to allow flight. So their absence has supported the conclusion that the Scansoriopterygids were not able to fly, which is already suggested by a lack of a distinct bird-like morphology in their skeleton. That is, until the discovery of E-Chi. In 2015, Zhu et al. Described this novel Scansoriopterygid and its very surprising morphology. It has a skin membrane attaching to its forelimb and a unique bone extending from its wrist, which has been dubbed the styloform due to its apparent functional analogy to the cartilaginous styloform of anomalurid and petaristine squirrels, like the one you can see down here. So suddenly it appears that there is good reason to believe that the Scansoriopterygids were experimenting with being airborne. And yet the debate still rages about the aerial capability of these little dinosaurs. Now, unfortunately, there isn't time to go through every aspect of the Scansoriopterygid skeleton and how it compares to fallen animals, but the key factor is that Scansoriopterygids appear to lack the shoulder morphology that would allow them to produce an upstroke akin to any other known flyers, because they essentially couldn't bring their arms together behind their back. And they also don't appear to have strong enough chest and shoulder muscles for a powered upstroke. But nevertheless, reconstructions of the Scansoriopterygids show these very visually convincing wings. So there remains this kind of nagging feeling that maybe these wings could really have facilitated flight. And this can be assessed quantitatively. Now, quantitative analysis has been used to assess flight capability previously in extinct vertebrates like bats, pterosaurs, and even in E itself. And then in October of this year, Takeshi et al. Published a very dramatic paper which stated that
just got a brief interruption of the video feed there. Can anybody else see Sophia? I think Sophia, you might need to click reconnect.
Is that use these simplified diagrams, which are all various models of the wing membrane and styloform.
So we'll just give Sophia a moment to rejoin there. I think she's having a couple of technical hitches. Do keep your questions coming in the chat. I can see there's a couple of interesting ones coming in. And I'll just go and make sure that Sophia can rejoin us.
Not sure what happened there.
Sorry, go ahead.
Yes, so where were we? Yes, so these are modeled arrangements for the styloform in the membrane. You can see the styloform is highlighted in blue here. And these are based on hypothesized reconstructions from Zhu et al.'s original paper. And I've also added an additional model which has the styloform entirely absent. These are the same general modeled arrangements that were used by Takeshi et al. These models that you can see here are entirely to scale, and they use bone measurements for each element directly from Zhu et al. And Wang et al., those who described these specimens. So from these reconstructions, wingspan and wing area were estimated. And then from those estimates, aspect ratio, which is wingspan squared over wing area, and wing loading, which is weight over wing area, were calculated. All of these values are really important in assessing flight ability. And these values for the Scansoriopterygids were then compared to a large data set of other flyers and gliders from the published literature. So what we have here first is a log-log regression, which shows log wing area against log mass for birds, bats, and pterosaurs. And it also includes Onychonicterus, the earliest bat, which is reconstructed as a flyer, and Archaeopteryx, one of the basal birds, which again is generally reconstructed as being flight capable. Here we can add in the E models. What you can see is that both the pterosaur and even more so the bat model increase wing area towards the range of flyers. Actually, the bat model lies directly on the regression line of pterosaurs, right next to Eudimorphodon. The Maniraptoran and frog models fall below even the glider regression, but they're still higher than Onychonicterus. So, and since that's considered a flyer, by that standard, perhaps those models aren't ruled out entirely of flight. Here are the Ambopteryx models. As you can see, they also fall below the glider regression. And actually, the bat and pterosaur models are barely any different from the model lacking the styloform because the styloform is so short in Ambopteryx. But now our most compelling result, this is a visualization of a linear discriminants analysis with all the previously included animals now classed as either flyers or gliders, and Scansoriopterygids introduced as a third group unspecified. This LDA has a classification accuracy of 98.8%. And the models which I've written in bold here, the pterosaur model, the bat model, and the no styloform model even, for E, are all classified as flyers by this LDA. None of the other models for either E or Ambopteryx are classified as either flyers or gliders. They are classified as a category of their own, but those three models are classified as flyers. So what we can see from the quantitative analysis is that the bat and pterosaur models of E are really promising candidates for powered flight ability, both improving over a model lacking a styloform. And that indicates the significance of the styloform in increasing aerodynamic ability. So then how can we reconcile these potentially flight-capable wings with a skeleton which doesn't have the features necessary for flight as we know it in other vertebrates? Scansoriopterygids don't have the upstroke-enabling shoulder morphologies of either pterosaurs, bats, or birds, so it's unlikely they were able to produce an upstroke analogous to any of the other flyers. But it is possible the explanation lies with Archaeopteryx. Archaeopteryx also lacks the shoulder morphology of modern birds, and its own flight has been much debated, but our analyses, as others have before, do support Archaeopteryx being flight capable, even though it lacks that shoulder morphology. So it presents a similar problem to the Scansoriopterygids. Several hypotheses have been put forward as to how Archaeopteryx might have been able to fly. In 2009, Nuds and Dyke suggested an active downstroke, passive upstroke model, which is similar to the way that round-winged birds like pigeons fly, essentially relying on vortices generated by the downstroke in order to produce an upstroke. There's another model which is known as the downstroke-only flap-assisted gliding model, the origin of which is slightly unclear, but it's usually attributed to Center 2006. And that essentially suggests an intermediate between powered flight and gliding, in which gliding behavior is aided by periodic powered downstrokes. Most recently, though, in 2018, Voitin et al. Found evidence that Archaeopteryx was definitely capable of intermittent powered flight, and they suggest a wing stroke that was not directed dorsoventrally as it is in modern birds, but instead anterodorsal posteriorventrally, diagonally across the body. And I would just like to expand on that model a little bit and say that a huge amount of power would be required to move that wing forward in the anterodorsal direction against the air resistance which is generated by the forward motion of the animal. And in fact, pushing against that force might actually counteract the motion of the downstroke. So instead, perhaps the wing is kind of folded against the body and moved forwards into position to begin the downstroke, kind of like a swimmer performing breaststroke. But regardless, any of these models could potentially describe a method utilized by the Scansoriopterygids if they were to get airborne. In fact, when we compare anatomically to the wings of other vertebrates, particularly those with membranous wings, the wings of E. and Ambopteryx actually have some advantages. Bats have extremely fine aerial control and agility because of their long fingers, which act as struts in their wings, which can be individually moved to alter sections of the membrane. The trade-off, though, is how fragile those long fingers are, with a high risk of tendon slipping. Bats have evolved an incredibly high number of sesamoids, which are floating bones, which in this case direct tendons over the joints, in order to mitigate this. Pterosaurs, on the other hand, only have one elongated wing finger as part of their wing, which reduces the risk of injury but also reduces their maneuverability. Scansoriopterygids also have one particularly elongated finger, which is most prominently involved in supporting the wing, but the styloform provides an additional stiff strut in the wing, allowing greater control of the membrane sections without sacrificing safety of the digits. Additionally, as seen in the previous analyses, the presence of the styloform increases the wing area to a more flight-optimal range. Bats and pterosaurs achieve this kind of wing area because the plagiopatagium, which is the main part of the membrane, attaches to their ankle. To facilitate that, the hind limbs of bats and pterosaurs are directed somewhat outward from the body, non-parasagittally, and that reduces their ability for cursorial movement. So increasing wing area via the styloform while avoiding ankle attachment allows the hind limbs to remain parasagittal and free for rapid cursorial movement, like running. So to summarize briefly, we've found some evidence to support the hypothesis that at least E.G. could fly with wings conforming to the bat model or perhaps the pterosaur model. If this was the case, E.'s wing stroke would not have been analogous to any known flyers, likely reliant on the downstroke. It's likely that E would not have been a particularly agile flyer, but thanks to added control from the styloform element, may have been perfectly able to navigate forests for short distances. In the future, it would be enormously advantageous to get an assessment of cortical thickness in the long bones of Scansoriopterygids, because this is known to differentiate between volant and non-volant archosaurs. Additionally, more complex, potentially 3D models which allowed simulation of the full wing beat cycle under varying conditions could undoubtedly add to our understanding. So with that, thank you very much for listening. Feel free to contact me on my social media or via email, and I am ready for any of your questions.
Super. Well, thank you very much, Sophia. We've got time for one quick question from Jane Pusey here in the chat. So Jane asks, what is the evidence that these are actually all bat-winged? If they've been found with feathers, why would their wings have been feathered and not just kind of a bare membrane?
Well, membrane has actually been found preserved in the fossils of E.G. and Ambopteryx and a very small part in Epidexipteryx. So we know that they did have a skin membrane attached to their forelimb, and they did have feathers as well. So Ambopteryx its name means kind of ambiguous wing. That's partly because its wing is it's a bird that's got kind of a pterosaur wing, but also it's got feathers and also a membrane. So they had both, but the key thing is they didn't have flight feathers. The feathers that they had would have been for thermoregulation or display or other purposes, not flight.
Super. Well, thank you very much. That's a very comprehensive answer to that question. So thank you so much, Sophia. We're going to move over now to our next speaker, which is Manabu Sakamoto. I will just prepare those slides. Okay, take it away.
Right, thank you. Hello. Thank you everyone for coming to my talk. For anyone who doesn't know me, my name is Manabu Sakamoto. I am currently at the University of Lincoln, and I have a passion for evolutionary biomechanics using phylogenetic comparative methods, some of which I will share with you today. So for this talk, I would like to talk about biforces and how reliable biforces are in extinct animals, in particular dinosaurs and mammals. So let's start off by asking why bite force. Bite force is the output of a biomechanical system. specifically the musculoskeletal system of the feeding apparatus. So it's the business end of the morphofunctional complex that comprises the jaw-lever system. As such, bite force is the physical outcome that is that this phenotypic complex comes under selection. So that is, selection works on bite force, and the associated phenotypes are indirectly selected. Then it follows that bite force likely correlates with dietary ecology, or hopefully we think so. Additionally, bite force is a single value trait, which means it can be represented by a single number for a species. This comes with a number of benefits when it comes to data analysis, since there's a huge repertoire of statistical analyses available for single trait responses. Lastly, but crucially, bite force can be measured, or if not, estimated. So it's quite a useful measure to have. Since dietary ecology isn't always available from direct evidence in completely extinct taxa, like saber-toothed cats or dinosaurs, bite force has often been used as a proxy for ecology. Or at least we can gain insights into the comparative biomechanics of feeding in extinct taxa and compare and contrast how various taxa have different biomechanical adaptations for biting. The hopes are that such comparative analyses will shed light on past ecologies. That begs the question, though, that how good are our bite force estimates in extinct taxa? Bite force estimates depend on muscle parameters, but since muscles are rarely preserved in their entirety in fossils, they have to be reconstructed one way or another. This involves some level of uncertainties and errors, particularly in muscle architecture, so that pennation angles and stuff like fiber lengths and cross-sectional areas. These parameters can really influence muscle force estimation. So we can make these lovely muscle reconstructions, like the one you see on with the T-Rex there, especially where they attach and how they orient. But the muscle architectures are more or less unknown. The effects of such uncertainties can manifest as variation in bite force estimates, as you can see from Bates and Falkingham studies, where they compare various estimates within their own work, but also across studies. So I'm just going to turn my video off here a bit. So they demonstrate here on the right-hand side that there's a fair degree of variation in bite force for T-Rex in their own study. So that's depending on parameterization of the muscles. But they also show that estimates from another study by Gignac and Erickson are substantially lower than their values. But is this a problem? How big of an issue is this? How severe are these uncertainties and discrepancies in bite force estimates across studies and parameterizations? Well, we don't know. So as far as I am aware, there hasn't been any wide-scale, statistically rigorous assessment of bite force estimates in extinct taxa. So to address this problem, what we can do is compare bite force estimates in extinct taxa against some null expectations. That is, we can generate an expected range of bite forces based on Brownian motion evolution and a prediction model using extant data. There'll be a model of bite force against a set of predictor variables, such as body mass, and this can be done over Markov-Chain Monte Carlo, or MCMC, to generate a posterior distribution of predictive values, or the posterior predictive distribution. So again, I'm going to turn my video off. So here, on the left-hand side, you've got the data set. On the right and in the middle, there's a phylogenetic tree that you can use. And then using this posterior predictive model, using phylogenetic information, we can generate this posterior predictive distribution. So we can then compare our biomechanically estimated bite force values against this expected range. And if the observed value fits within the posterior predictive distribution, then we can conclude that our biomechanical estimates are not significantly different from null expectations. But if it falls on the periphery of this distribution, then we can conclude that it is significantly different from expected. So interpretations for significant deviations can be one of two things, or maybe there's none, but I can think of two. One is that the bite force of the taxon is genuinely an outlier, so as in it is the result of a real adaptation. Two, that the bite force estimate is likely wrong. So to do this, I use the regression To do this, I use the phylogenetic posterior predictive modeling approach I just showed in the previous slide. And I fitted a phylogenetic regression model on, sorry, I'm on the wrong slide, sorry. Regression model on data in extant taxa. So with biforce as the response variable, and using the following variables as predictors. So body mass, skull width, the biting position, because biforce can vary depending on where on the tooth row the biforce is estimated. And this is basically used as a controlling factor, and grouping structure. So this is whether taxon is a bat or a finch or not, because these two groups have steeper slopes than the rest of the taxa. So then this regression model is fitted through MCMC, as I said before, and a posterior distribution of predictors is generated using Bayes traits. Right, so this is the slide I was actually, I just kind of went ahead, but so then before I actually predicted the biforce in extant taxa, I validated the model through leave one out cross validation, or LOOCV. In this validation approach, the model is
Just give Manabu a minute to reconnect there. Thanks for bearing with us, everybody. We're just going to see if Manabu is able to rejoin before resuming. Okay, so it looks like Manabu's just left the session, so I think he might be having some technical issues. Hopefully he's now rejoined, so if you are happy to restart, Manabu.
hello, can you?
Yes, yeah, we can hear you again now. Sorry about that.
Computer, I don't know why it just died, and I had to restart for some reason.
Are you on the right slide? Can you see everything okay?
Yeah, can you see?
Brilliant. Yeah, we can see it and hear you absolutely fine.
Yeah, sorry, I don't know why my computer just died. Yeah, I was saying that out of the 188 taxa in our training set, 14 taxa had biforces significantly different from the posterior predicted distributions, which is shown here in pink. And fortunately, all of these 14 taxa are known to have exceptional biforces, such as the aardwolf with a very low biforce for a hyaenid, or the finches, which are very famous for having undergone adaptive radiation in Fiji ecology. So from these results, we can safely say that biforce can be predicted accurately from the posterior predictive model. Using this sorry, yeah, I got some technical issues. Sorry. Yeah, okay, so that's fine, sorry. So then using this posterior predictive model, I generated posterior predictive distributions in 37 extinct taxa. So these are shown in these two shades of pink. These include mammals like saber-toothed cats and a couple of marsupials, but also archosaurs, namely dinosaurs. So observed biomechanically estimated biforce values generally fall within the posterior predictive distributions and significantly different in only 4 taxa, shown here in the dark pink. These 4 are 2 species of saber-toothed cats, Plantosaurus and Stegosaurus. So ignoring the dark pink for now, biforce estimates are as expected for body size, skull width, and evolutionary history in a majority of extinct taxa tested here. So then what about these 4 outliers? Well, they can be explained pretty well. For instance, saber-toothed cats have been known to have extremely small biting muscles and thus weak bites. And Plateosaurus and Stegosaurus, on the other hand, have very extremely small heads compared to their massive body sizes. So here's Matt Dempsey's awesome reconstruction of Stegosaurus. I'm going to turn my video off for that. So, and you can really see how just ridiculously small its head is in relation to its body. And I think there was a talk earlier today about a reduction in head size in herbivorous dinosaurs generally. So this is kind of something that you would expect with herbivorous dinosaurs. So even if the Bifors was accurately estimated for these taxa with the amount of jaw muscles available, in particular in Stegosaurus, they would have had exceptionally low bite force compared to what we'd expect given its body size. Whoops, sorry. Yeah. Lastly, I just wanted to comment on that issue of the discrepancies in bite force between the authors. I mentioned earlier. In the T-Rex example I mentioned earlier, biomechanical bite force estimates are actually not significantly different among studies, according to my analyses. So bite forces from the Guignac and Erickson study, my own study from before, and Bates and Falkingham all fall within this within the 25th and the 75th percentiles of the distribution. So that's really like smack right in the middle of that distribution, as you'd expect given its body size and skull width and stuff like that. And so far as comparative and evolutionary biomechanical studies are concerned, these discrepancies thus are is very negligible. It's very small. So basically, they're within expected range of error. So the only bite force estimate that is significantly different, however, is one that wasn't estimated biomechanically, but from an extrapolation of a regression model fitted on a data set that includes incorrectly double values. And so that's the 250,000 Newton estimate from the Mears 2002 study, which is an order of magnitude higher than any of the other estimates you can see here. And this value sits right on the threshold of the significance area. So that is that only 5% of the posterior predictive distribution are higher than this estimate. So in conclusion, biomechanical estimates of bite force in extinct taxa are as expected given body size and skull width and evolutionary history. And the error we observe in extinct taxa compared to that inherent in extant taxa. And differences owing to parameterization or across studies are relatively small on a comparative macroevolutionary scale. And lastly, provided that general muscle dimensions can be reconstructed, bite force estimates can be treated as being reliable as that measured in extant taxa. So in other words, your bite force estimates are pretty good. So don't worry about drawing macroevolutionary and paleoecological conclusions from them. So that concludes my talk, and I just wanted to leave you with some shameless plugins, and I'll take any questions if you got any, and thank you for listening.
Super, thank you very much, and well done there for bearing with us through those slight technical issues. You were incredibly well timed, so thank you very much, Manabu. So we've got a question in the chat here from Eli Amson, who says, really nice analysis, but why assume Brownian motion in the evolution of this trait?
So what about the Brown?
Why are you assuming Brownian motion?
Oh, well, why assuming Brownian motion? Because Brownian motion is basically the least of the least it makes the least amount of assumptions, and previous kind of assessments and comparisons of the other models tend to show Brownian motion is very good for things like bite force and body mass. So body mass evolves under Brownian motion for a lot of things and in a lot of groups, and bite force is very tightly correlated with body size, and therefore it really for the most part it's not really non-Brownian motion processes don't really come into it, and you don't really have like better model fits for that.
Super, great. We've got time for one more quick question here. So this is actually from Sophia. So this is brilliant talk. What are your thoughts on the efficacy of Thomason's dry skull method? So
Thomason's dry skull method is really well known for being underestimating. So it can't really get the really the muscle cross sections correctly, but there are correction factors that are known very really well. I think it was like a scaling factor of 1.3 or something. It's like you just multiply the muscle cross section area by that amount, and you get a fairly accurate bite force compared to in vivo estimates. So you can get around with it, and especially if you're using it in a comparative sense, like all your data is coming from dry skull, then there's absolutely no problem with using that.
Okay, so it's just a comparison between different data sets. Super. Well, thank you very much. That was really interesting.